In your cells, UPS isn't where you go to take packages to ship. Instead, it's where you go to take proteins to degrade. So the ubiquitin proteasome system is the way that our cells can tag proteins with another protein called ubiquitin. It puts these little chains of ubiquitin onto a protein, and then that serves as a signal, and then it gets taken to this like protein shredder called the proteasome. And this is a way that our cells can get rid of proteins that are damaged, that aren't wanted. Maybe they were made wrong. Maybe now it's just a different developmental time and you don't need it anymore. So the UPS is super duper important, and here's the biochemistry of how it works.
Okay, so there's a bunch of things that we're going to go over, but let's start with the idea that this is being used by our cells all the time, and it's super duper important. And it's also, especially more recently, become an important experimental tool. You might have heard of oxygen-inducible Degroms, AIDs, or PROTACs, these like proteasome targeting chimeras, various things like that, that take advantage of our cell's ability to degrade proteins that aren't wanted, or in the case of these like more artificial methods, to degrade proteins like that we don't want anymore on like a specific level. Instead of preventing them from getting made, we destroy them after they're made. So this is going to be an important experimental tool as well as potentially therapeutic if you're able to target proteins that you don't want to this proteasome. But first we need to understand, okay, well, how does this proteasome actually know how does things actually get targeted to this proteasome? And that involves this ubiquitinylation through this through ubiquitin ligases. And so there are different ubiquitin ligases, but they come in like these three parts. And so there's this E1, aka ubiquitin activating enzyme. This is going to get the ubiquitin ready to go using some energy from ATP, as we'll see. Then there's an E2, which is the ubiquitin conjugating enzyme. So once it's activated, then it gets passed on to this E2. And then this E2 is going to take it to the E3. The E3 is going to be what's actually attached to the substrate, to the thing that's going to get ubiquitinated. Some people say ubiquitinate, some people say ubiquitinylate, and I don't know what the consensus is because I've seen it both ways all the time, and I never know. But anyway, it's going to then get E3, the ubiquitin, depending on the type of ubiquitin ligase, I'll get into this, but there are different types. Some of them go directly to E3, or some of them, the ubiquitin gets passed off from E2 to the substrate directly. The E3, though, it is holding the substrate, but not the ubiquitin. Other times it gets passed to the ubiquitin ligase, so it gets passed to E3 and then E3 puts it on. One of these ubiquitins isn't typically enough to make something a target for the proteasome. Instead, something needs to be like polyubiquitinated, which is good because if it just got like one ubiquitin, maybe that was like an accident or something. It's not like a strong link. And so you don't want that to get accidentally like destroyed, and so you typically get polyubiquitination. And so ubiquitin is just this little protein, and it's about 76 amino acids, I think, and it gets linked up to the protein, and then you can have it gets linked into these chains. So it can get linked up at the C terminus, or sorry, the C terminus of it gets linked up to a lysine or to the N terminus of a protein. So we'll get more into this later, but it's forming a covalent bond. So this is a strong bond. It's an isopeptide bond. So it's a peptide bond because like the peptide bonds in our proteins, it's got this amide group. Don't need to know the details of this right now, but this is an isopeptide bond. It's hooked up in a different way. Instead of being hooked up directly to the protein backbone, it's hooked up to the side chain of the lysine or to the N terminus. So it's a kind of cool linkage. But anyway, the important thing to know is that it's like a strong linkage, so it's going to get stuck on, and then you can get chains of these ubiquitin put on. You could also get ubiquitin. So in addition to getting it just like on the N terminus, you can also get it to other lysines in the protein, and therefore you can have like branching of ubiquitin, and there's a bunch of work that's being done to try to figure out what these different like ubiquitin codes might be. So cool things like that. But anyway, let's get back to the story and we'll get more into the deal with the ubiquitin part later. So we have these three parts to our ubiquitin proteasome system, or I guess we have four parts. We have the proteasome as well, but we have these three parts to the ubiquitin ligase complex. So we have this E1. There's only two of these we know of. E2, there's about 40 of these. And E3, there's about 600 of these. So this is really where the specificity is going to come in. And this is what's actually going to bind to the substrate. And this allows you to target a bunch of different substrates using the same system. So our cells have kind of like specialized in or devoted their resources to specializing these E3s, and so you don't have to worry about this part. This is going to be, I mean, like you might have to worry about it, but your cells just have to get to use the same thing for all of these E3s. But if they focus their energy on just making sure these E3s are really specific, then they don't have to worry as much about the specificity of these. But there's still a couple of main classes of these ubiquitin ligases. So this is just some stuff from my blog, just for my notes, and so that you guys can see that I have a bunch more on my blog if you want. So yeah, another thing is that these can be deubiquitinated as well. So that's another thing, instead of just having the check that you have multiple ubiquitins, you can also deubiquitinate things if you decide that you don't want to destroy them. And so there's these deubiquitinating enzymes or dubs that can like remove them. So there are two like kind of big categories, or I guess three, I don't know, of these ligases. So ring ligases, they don't pass off to E3 before they go to the substrate. Instead, the E2, this E3 brings in the substrate, and then the E2 just passes it directly from E2. The HECT and RBR ligases do the final transfer themselves. So they have to first transfer it from E2 to E3 and then onto the protein. So let's go through these different steps. So E1, this is where we're going to do the activation. So we need to activate it. We need to give energy for this process to go, because the ubiquitin is not just going to like plop right onto the protein, in addition to having to bring things together, which is what we have like the E3 and all this stuff helping bring all of these things together. And so these enzymes, so these reaction mediators, they're making things actually happen, but they're also going to need some energy to make it so that the ubiquitin will actually like want to go onto a protein. So to do this, we need energy. We get this energy from ATP. So ATP is just like the cellular form of energy, one of the main forms of energy. So these three phosphate groups are negatively charged, held close together. You can think of it as like a clamped spring. Those negatively charged phosphates, they really want to get apart from one another, and so it takes like chemical energy to hold them together. And so energy cannot be created nor destroyed. So if you are to break those bonds, then you're going to release the energy that was taken to hold the spring together. And so when you then add on to this ubiquitin, when this ubiquitin attacks this ATP, now it kicks off this pyrophosphate, these two phosphate groups, and then those can hydrolyze so they can break apart, and you get even more energy because you're breaking a bond, and then you're getting this entropy benefit. Basically, things like to be random, and so you have more randomness. So this can be really great. But anyway, this is going to get adenylated. The ubiquitin is going to get adenylated. So remember, ubiquitin is this little protein. We're going to add we're going to adenylate it. So we're going to, instead of just adding a phosphate group, we're going to attack it, and we're going to take this adenosine phosphate onto here. And so now this linkage is going to be more vulnerable to attack. So basically, the reason why this isn't going to be that great at attack, this wouldn't be very attackable. This makes it more attackable because you're making a better leaving group. So when you have these reactions, when something is going to come in and we want to attach it to here, basically in order to attach it to this carbon already has 4 bonds to it. Carbon can only have 4 bonds total. So we're going to have to kick something off in order to get that linkage. If we did that here, we'd be kicking off like an OH, which isn't that great of a leaving group. But if we do it here, we're kicking off this adenosine phosphate, which is a much better leaving group. We have a much easier chance of breaking this off. So this is going to help like make it so that we can do that transfer. And you'll see this sort of thing a lot in various biochemical reactions that I've talked about before. Okay, so this is gonna like kind of like bribe the E1 to bring to bind to the ubiquitin. And so now after we've activated the ubiquitin, so we haven't even gotten the ligase in here yet, we haven't even gotten any part of like the E1 or anything. Now we're gonna bring in the E1, and so now this is more attractive to the E1, and E1 can do that attack that we were talking about, and it's going to do this attack through a cysteine. So this amino acid cysteine, and it's going to be able to, because it's nucleophilic, basically it wants to seek out something electrophilic, or and so it wants to have basically I don't want to go too much into detail here, but I have more posts on those other places. But basically, that bond is going to be electrophilic, and so we have this nucleophilic cysteine. What it's going to be able to do is, if I can find the slide, it's going to be able to attack that bond that we just talked about, break that off, release the adenosine monophosphate to AMP, and now you have this ubiquitin conjugated to the E1. And so when you have this conjugation, you often see it like represented with this like squiggly line. Okay, and so we say, yeah, this is like conjugated, it's like covalently attached, but it's gonna also be like passed off, as we'll see. And so it's going to get passed off in a trans thioesterification. So this is an ester bond, a thioester bond. And so an ester bond would be when you had an oxygen in between two carbons, and one of the carbons had a carbonyl, that would be an ester bond. This has a sulfur, and so it's the sulfur version of it. It's a thioester bond. We have a sulfur or something, we put the term thio. So this is a thioester bond. Now we're going to do a trans thioesterification, which means we're going to basically make a new thioester bond on a different thing. We're going to do it like swap it out. So we're going to take that ubiquitin that's attached to E1, and we're going to swap it to E2. Okay, so we do the swap to E2, and now E1's done its job, and it can go and get another ubiquitin to bring and stuff like that. Now E2's job is it's going to bring the ubiquitin to E3, and either directly pass it off to the substrate or pass it off to E3, which passes it off to the substrate. And E3 is going to be the one that's actually bound to the substrate. So the E3, with the substrate to be tagged, the ubiquitin is ubiquitinated E2, and then these are going to come together, and the substrate is going to get passed off to the substrate, either directly, in the case of these, the ring E3 ligases, is going to get passed directly from to the protein, or in the case of these HECT and RBR E3 ligases, they're going to take it for themselves first. So you get another trans thioesterification, and then it gets passed off to their substrate. As to what these actually look like, here's an example of a ring ligase. So you can see that you have, this is a colon ring E3 ligase, and you can see here's our E2. We have our substrate bound to E3. And then we have, you can see that the pass off is going to occur. So it's pretty cool stuff. And so let's talk about how, though, this substrate is actually going to get recognized by this E3 ligase, because that's a really important thing, because that's where all of our specifications going to come in. That's where we're going to know, okay, like, well, how do we actually know what we're going to be targeting, and how maybe can we target things that aren't normally targeted but that we want to get rid of. And so basically we have to understand, okay, so this is why lysine is able to form these bonds, but our proteins have a lot of lysines in them, and so how is it that they are able to form these linkages? And so we have the N-terminus, as we talked about before, and so every protein has one of those. That is the potential linkage point. Then they also have lysines throughout the protein. If you have there are basically certain amino acids that are going to be more recognizable by the proteasome. So these can serve as these things called degrons. So degrons are basically the signals for the proteasome to degrade things. There's like these serve as these recognition signals, and they're going to be recognized by a part of E3 called a recognance. That's just a part of E3 that recognizes the degron. And so this is going to be this is important because the E3 has got to hold on to that substrate, while E2 keeps leaving and coming back and bringing in new ubiquitin. So we need this to be a strong bond, and we also need that to be really specific so that we're not just ubiquitinating anything. And so some of the methods, like experimental methods that are used, are actually to get this E3 to bind to the substrate and that sort of thing, even if it wouldn't naturally. But what we're talking about with the naturally is based on these things, these degrons, so these naturally occurring degrons. So these often occur at the end, and so we have this like N-terminal rules. And basically, if there are certain amino acids at the end of the protein, then it's going to make it more. It can serve as a degron. These sequences, though, aren't always at the end of the protein. You have like a hidden degron within the protein, but if that protein gets cut, well, now you have a new end. And so certain amino acids can serve as degrons if the protein gets like cut. And so proteins can get processed in a way that then makes it easier for them to get degraded. Proteins can also have degrons that are like always there, but they're hidden. And so this can be a developmental strategy that you kind of have a binding partner kind of binding on the part that would serve as a degron and protecting it so the proteasome can't see it, and therefore it doesn't get degraded until like the developmental stage when the partner then leaves and the protein degron is exposed and the protein gets degraded. So that's a cool strategy that's used with things for like our DNA replication and various cell cycle things a lot. Proteins can also just be bound to other proteins, and then those proteins get degraded. Yeah, so there's a bunch of different things that can make it so that degrons are hidden or not always there. For some amino acids can serve as degrons like directly. Others get modified first. And so there's basically, we can think about, there's these different pathways, and I have more on this in my blog post. There's like this ACN degron pathway, this ARG-N degron pathway, this pro-N degron pathway, and again, N because we're talking about the N-terminus. And some are primary, so they can be bound directly by ARG-N recognants. So these are, remember, recognants. Those are the parts of the E3 ligases that can recognize the various things. Others that are secondary, that they get one modification will make them into a degron. Some are tertiary, so you can get two modifications to make them into degrons and various things like this. If you want to know more, there's this really great article showing the structural basis for how these degrons are recognized by these recognants. So these parts of the different protein, the E3 ligases that then recognize them. So some really cool structural work by Alexander Vashovsky in his lab. So very cool. Here's a good review paper. Okay, so that's the naturally type stuff, but we can also use this experimentally. Say if we want to get rid of a protein in a cell, maybe it's a protein that is essential, so we can't just get rid of it all the time, or the cells wouldn't grow at all. What we can do is we can use something called an auxin-inducible degron to induce degradation of it, like on command, by adding a molecule. That will then make it so that this protein is a substrate for this, for the ligase that it wouldn't normally be a substrate for. So it's like a neo-substrate. So in nature, this happens through these hormones, these auxin-inducible degrons. So auxin is like this plant hormone or this class of hormones. And so we can use this synthetic auxin is often used as NAA, or a natural one is IAA. Basically, there's this FBOX protein. It's going to be like the E3 ligase that determines the specificity for this. And there are different FBOX proteins. There's this one called tier 1, that's a plant one, that only binds to the two auxin-inducible degrons when auxin is present. So auxin kind of like acts as a glue that holds them together. And so when the auxin is absent, the protein goes about its business, but then the plants use this auxin to induce a signal that then gets those proteins targeted, and then it gets ubiquitinated, taken to the shredder. we can do things experimentally where, so our cells say don't have this plant protein, but if you get the cells to express this FBP, then you can use it to induce a protein to get degraded. If you also put the tag, an auxin-inducible degron onto a protein. So you make this protein, you put on a little extra information. So this would be, you can either do it through like endogenous labeling, where you actually like edit the genome, or express like through a plasmid or something. Where you basically put this auxin-inducible degron onto the protein. The protein goes about its business, but now when you add the auxin, or the synthetic auxin, then the protein gets degraded. And so in addition to being able to use this if your protein is essential for the cells, you can also do it to stop a protein at a specific time, various really cool things like that. So that's like an experimental system where you're actually having to express these things in a artificial way. And you have to actually tag the proteins in order to degrade it. So this wouldn't be like a therapeutic strategy, but there are therapeutic strategies. This is things called PROTACs, so proteasome targeting chimeras. And so basically they're molecules that are inspired by these, but instead of using auxin, or an auxin analog, and instead of using these plant ligases, they take advantage of our own cell's ligase and try to build a molecule that will bind to a substrate and bind to the ligase, bring them together, and that way degrade a protein of interest. So there's a lot of cool work being done in that area, as well as work being done to understand our own proteasome systems, how they can become like overloaded in certain diseases, how they can be used in how they can be coupled with translation. If protein making goes awry, you can basically get rid of that partial protein on demand. There are various ways in which the proteasome is involved that are really important in all a bunch of cellular processes. And so, yeah, so hope that helped you understand these ligases, ubiquitin proteasome systems, and Degroms, all that cool stuff. Yeah, so hope it helped.